everyone. This is week 4 in our final week on our focus on carbohydrates, and we'll be talking about carbohydrate metabolism. Okay, our objectives for this class are to list and discuss the individual reactions of glycolysis, both aerobic and anaerobic, discuss the metabolic fate of pyruvate, lactate, and ethanol, discuss the individual reactions of the TCA cycle and of the hexomonophosphate shunt, also known as the pentose phosphate pathway, and discuss gluconeogenesis and its substrates. Okay, so the predominant pathways of glucose metabolism vary according to cell type and physiological demand. So what is our body going through at the time? Are we fasting? Are we exercising? Did we just eat a big meal? What kind of tissues are we supplying energy to? Do they have mitochondria? Can they go through oxidative phosphorylation and so on? So the metabolic pathways of carb use and storage include glycogenesis and glycogenolysis, which we talked about last week. And then this week we'll be focusing on glycolysis, the TCA cycle, the hexomonophosphate shunt and pentose phosphate pathway, and gluconeogenesis. Okay, so the liver plays a central role in the body's glucose homeostasis. So in hepatocytes, glucose can either be oxidized for energy via the TCA cycle and electron transport chain. When we have them in glucose in abundance, our liver can store the glucose as glycogen, and it can also provide carbons for the biosynthesis of other macronutrients, including fatty acids and amino acids. So if blood glucose is low, I think this is a review for all of us, hopefully, the liver is able to release glucose by degrading its glycogen, and it's also able to produce new glucose, especially for tissues that rely exclusively on glucose, and to maintain blood glucose homeostasis. Hepatocytes can utilize glucose also for production of NADPH and ribose 5-phosphate through the hexomonophosphate shunt or pentose phosphate pathway. And we're going to be talking about that moving forward. Okay. So in the skeletal muscle and heart, glucose can be completely oxidized for energy, again, via TCA and electron transport chain, or if glucose is present in abundance, it can be stored in the form of glycogen. However, glucose in the muscle cannot contribute to whole body glucose homeostasis. And you'll remember from previous lectures that that's because muscles lack G6 phosphatase, so that they're not able to convert G6P back up to glucose that could be released back into the blood. So instead, it needs to either be oxidized or stored. There are different needs depending on the type of muscle. So for instance, the heart has a continuous energy need for regular contractions, while the skeletal muscle has more variation in its needs depending on how much exertion. You can see over here that there's different types of muscle, and when we talk about smooth muscle, that's the muscle in the GI tract, the uterus, and the blood vessels. Okay, so in the adipose tissue, glucose can be partially degraded by glycolysis to provide glycerol for triacylglycerol synthesis. Sorry about that. I'm going to take a little drink here. And triacylglycerol is just kind of the chemical name for triglycerides. So if our carb intake is high, glucose is metabolized to acetyl-CoA, and the acetyl moiety of the acetyl-CoA can be channeled for fatty acid synthesis for storage. So this is just another way that we're able to use glucose to store energy other than glycogen. If we have a need for energy, adipose cells release the metabolic fuel in the form of fatty acids to circulating blood supply. So what about in our other cells? Well, our brain is one of the tissues that's completely dependent on glucose for needs and is capable of oxidizing glucose to carbon dioxide and H2O via the glycolysis and the TCA cycle. Now, a couple of little red flags may be dinging because you're thinking, hey, what about ketone bodies? Now, the brain is able to utilize ketone bodies for energy, but our ability to degrade and excrete ketone bodies is somewhat limited. So in most people, if we have this buildup of ketone bodies without enough glucose, it has a negative impact on the brain. We know this as ketosis, which can ultimately lead to either coma or death. So we also have other tissues that are dependent on glucose for needs. And these are the tissues that lack mitochondria and therefore are non-oxidative and unable to utilize the energy from a complete oxidation of glucose. So these cells include the cells that don't have a mitochondria. So for instance, red blood cells. So red blood cells rely on glycolysis, the first part of glucose degradation, but then are not able to fully oxidize glucose through the TCA cycle. So other cells without mitochondria or that have a limited blood or oxygen supply and therefore cannot undergo the TCA cycle and oxidative phosphorylation include the cornea, lens, and retina in our eye, leukocytes, white muscle fibers, cells of the testes, and the renal medulla. Okay, so here is the steps of glycolysis. Hopefully you have a pretty big screen there. So we can see here, we're going to go through all of these steps, but I just want to point out a couple of things. So this one says hexokinase as the first enzymatic reaction. This also refers to glucokinase in the liver. And one thing I do want you to note is that here we have the breakdown of glucose until step number 4 or 5 here, when fructose 1,6 biphosphate breaks into 2 different molecules that can be converted back and forth until they reach equilibrium. And therefore, and just this G3P, the glyceraldehyde 3-phosphate, so we end up with 2 of that, because even though we're converting it to the dihydroxyacetone phosphate, that can be converted to the G3P here. So we end up with 2 molecules of G3P and ultimately 2 molecules of pyruvate. So for every I should say moles. For every 2 moles of glucose, we end up with 2 moles of pyruvate. So really, this first part is happening once, and the second part is happening 2 times for every mole of glucose. Okay, so, oh, and I should go back and say that after pyruvate, the molecule enters the TCA cycle, where, and we can see here in glycolysis, we actually use ATP, but once we get into the TCA cycle, we only produce energy and not use energy. Okay, so I'm going to go through the glycolysis reactions. So reaction 1 of glycolysis is the stage where glucose is phosphorylated. So we actually have an investment of ATP ending in ADP, and it's phosphorylated to G6P. So we have the added phosphate group on here. This is ATP dependent, and it's catalyzed by tissue-specific isozymes known as hexokinase. And the word isozymes just means that these are enzymes with identical function but different structure. So one type of hexokinase is glucokinase, so that's specific to the liver. So phosphorylation of glucose accomplishes 2 goals. First, it converts glucose to an anion that can no longer be transported out of the cell. It's trapped in the cell um, in order to undergo oxidation. So cells lack the transport systems to move phosphorylated sugars out of the cell. In addition, the biologically inert glucose becomes activated into a form capable of further metabolism. And we talked earlier in a previous lecture about how this is the first step in multiple reactions, including not only glycolysis, but also reactions involving glycogen. Oh, and I should also say down here, I said that glucokinase was specific to the liver, but it's also present in the kidneys. So glucokinase allows the liver to buffer blood glucose. So when blood glucose is high, the liver can preferentially trap and store circulating glucose. So think of when we've just consumed a meal and our postprandial glucose is high, then glucose is significantly active. So we talked about in a previous lecture how glucokinase is really only activated when we have high blood glucose. Now, when blood glucose is low, such as after periods of fasting, the liver and the kidneys do not have activated glucokinase. So they don't use the meager glucose that's available instead they, because they themselves are not highly dependent on glucose. Instead, you can think of, they leave the glucose for the tissues that are much more dependent on glucose, such as the brain or cells like red blood cells. So during fasting, the liver supplies blood glucose via gluconeogenesis. So if we think about it, we're not breaking down glucose when we, so the liver provides, so when we have low blood glucose levels, these are insufficient to activate glucokinase. So when we're high, glucokinase is active. When we're low, it's not enough to activate glucokinase to break down glucose. So moving on to reaction 2, which is the isomerization of G6P to F6P. So that makes sense. It's an isomer. We have the same molecule, or I shouldn't say same molecule. We have the same atoms, but in a different configuration in this reaction, and it's catalyzed by phosphohexose isomerase. So that's shown here. And then we have reaction 3, which is ATP dependent. So again, we're having an investment of energy at this stage in the oxidation of glucose. And the reaction includes the conversion of F6P to F16BP. So again, we have one phosphate here, we have a transfer of the other phosphate so that we have two phosphates. And this is catalyzed by phosphofructokinase 1, or PFK1, which I will refer to it as moving forward. Okay, next we have reaction 4, in which fructose 1,6-BP is converted into 2 carbon products. Okay, so this is that split that we talked about on the earlier slide that had the overview of the whole pathway. So it splits into 2 molecules, the DHAP and the glyceraldehyde 3P. Okay, so we break down this molecule into 2 smaller carbon molecules, and these can equilibrate. Okay, so it has to be in this form, the glyceraldehyde 3P, in order to continue on the glycolysis pathway, but DHAP is able to convert to glyceraldehyde 3P. Okay, and this is catalyzed by the aldolase enzyme, and the reaction can go in either direction. So this reaction is utilized for both glycolysis, the breakdown of glucose, and gluconeogenesis, which is the production of glucose. But let's just focus on glycolysis here. And then reaction 5 is just the conversion of dehappened glyceraldehyde 3P. Okay. And our next reaction, this is kind of the second phase of glucose catabolism. So in our first phase, we used ATP. That was the ATP investment. And then in the second stage, we're actually yielding energy to produce ATP and NADH. So this is the first energy yielding step. So in this step, we have the conversion of the glyceraldehyde 3P from the previous step, and then we have it's converted to a 1,3-biphospholglycerate with the enzyme glyceraldehyde 3P dehydrogenase. So we're actually producing NADH at this step. Okay, then we have reaction 7, which is the conversion. Ooh, you can't really see it right here, but very well. But the conversion of 1,3 BGP to form ATP and 3-phosphoglycerate. So again, this is energy producing step. Okay, and then we have our last steps of glycolysis, which are aimed at converting the relatively low phosphoacyl ester of 3-PG to the high energy form, and then harvesting the phosphate of ATP. Okay, so in reaction 8, we have the conversion of 3-PG to 2-PG. So we're getting rid of one of the phosphates and we're utilizing that to produce ATP. In reaction 9, we have the conversion of 2-PG to PEP. And then in reaction 10, which is our last reaction, we have PEP converted to pyruvate, catalyzed by pyruvate kinase. And again, this is an energy producing step, and pyruvate is our final product of glycolysis that can then go on to the TCA, to be converted to acetyl-CoA to go into the TCA cycle. Okay, so under aerobic conditions, the pyruvate is enters the mitochondria and is converted to acetyl-CoA to go on to the TCA cycle. And this reaction is catalyzed by pyruvate dehydrogenase. So this makes sense because we are taking the hydrogens from the pyruvate, adding them onto NADH. So this is being dehydrogenated. And then this is an energy producing step. Under anaerobic conditions, the pyruvate is converted to lactate, and this is catalyzed by a lactate dehydrogenase enzyme. So if there's not enough oxygen in the cell, or the cell is a non-oxidative cell like a red blood cell, the pyruvate is converted to lactate. And this provides the cell actually with a mechanism of oxidation of NAD to NAD+. So then we can use that NAD for the reaction we talked about in the last slide of the pyruvate dehydrogenase. So it's a mechanism to kind of get the substrate of NAD to be used in further reactions. Now, when lactate is produced, it's then transported out of the cell into circulation to other tissues that are oxidative and can undergo oxidative phosphorylation, such as the cardiac, muscle, and the liver. It's then in those tissues, it's then oxidized to pyruvate by lactate dehydrogenase to be further oxidized via the TCA cycle. And then if we have more than enough energy, then the pyruvate is converted back to glucose instead of being oxidized for energy. So, however, aerobic glycolysis generates much more ATP per mole of glucose compared to anaerobic glycolysis, and we'll see how that exactly works out as far as ATP production. But aerobic glycolysis is much more desirable because we can get a lot more energy from it compared to anaerobic glycolysis. So mammalian cells have actually two distinct types of subunits on the lactate dehydrogenase enzyme. So there's an H-type and an M-type subunit. And the H-type subunit predominates in aerobic tissues, such as the heart muscle. So we talked about earlier that the heart muscle has a constant need for glucose, or for energy, because it's constantly contracting and working. So in these type of tissues, there is a low affinity for pyruvate, and it's inhibited by high levels of pyruvate. And then in the M-type subunits, which predominate in tissues that may be more likely to undergo anaerobic conditions, such as the skeletal muscle when we're undergoing extreme exercise, they have high Km for pyruvate, and it's not inhibited by pyruvate. So this suggests that the H-type of lactose dehydrogenase is utilized for oxidizing lactate to pyruvate in muscles such as the heart, and the M-type, the lactate dehydrogenase, is utilized for converting pyruvate to lactate, because that's more necessary in skeletal muscles. That's just kind of a side note. I wouldn't worry too much about it, but just for kind of interest's sake and to tie some points together. Okay, so how is glycolysis regulated? So first, I'm just going to kind of leave this figure up while I kind of talk through it. So the most important regulator of glycolysis and gluconeogenesis is fructose 2,6 biphosphate. So we see that here. And it's interesting because this molecule is not an intermediate of any of the pathways. So it's produced by other pathways, but is used to regulate gluconeogenesis and glycolysis. So when I say a regulator, this type of regulator involves the binding of an effector molecule to a specific site on an enzyme in order to initiate the impact that we want it to have. So the major sites of regulation of the F26 biphosphate are on this PFK1 enzyme and also on the F16 BPase enzyme, both of which catalyze the conversion between fructose 6-phosphate and fructose 1,6-phosphate, but in different directions. So PFK1 drives the reaction down, whereas F16 BPase drives it up. And this makes sense because we can see PFK is a kinase, which adds the phosphate on to drive this down, and biphosphatase takes the phosphate off and counters the action of the kinase and drives the reaction the other way. Okay, so F26BP is catalyzed by the bifunctional enzyme that is PFK2 and F26BPase. Okay, so in the non-phosphorylated form, the enzyme is known as PFK2, and it catalyzes the F2-6BP synthesis. Okay, so so the kinase adds the phosphate on and makes it active, and it results in the stimulation of PFK1 and the inhibition of F16BPase, and therefore drives the reaction down. However, when it's phosphorylated, the enzyme is the F16BPase, and or I'm sorry, the yes, F26BPase, sorry, and the enzyme no longer exhibits the kinase activity of the PFK1, and then it drives the reaction down and results in the hydrolysis of F26BP to F6P and an inorganic phosphate. Okay, so this, it shows it this way, but really what it's doing is driving the reaction back up to this fructose 6-phosphate. And these next two slides are what I just talked about, but you can have them for your reference. Okay, so now let's move on to ethanol metabolism. So animal cells contain alcohol dehydrogenase that helps us to break down ethanol. So in this reaction, ethanol is oxidized to acetaldehyde and then oxidized to acetate. And acetaldehyde and acetate are toxic molecules that lead to the side effects that we associate with excessive alcohol consumption, such as a hangover. Okay, next we're going to move on to the TCA cycle. So we've talked about this cycle some, but I want to go into more details about the actual reactions that are occurring in the TCA cycle. Okay, so if we kind of look up here, remember we had the pyruvate that entered the mitochondria and was converted to acetyl-CoA, and that's how we, the substance or the molecule that we use to kind of start this TCA cycle. Okay, so first we have reaction 1, which is catalyzed by citrate synthase. So we have the acetyl-CoA plus the oxaloacetate. So hold on, that might be confusing, so I'm going to go back. So oxaloacetate is produced in this last step of the TCA cycle, and then that's what's combined with the acetyl-CoA. Okay, and then again, it's catalyzed by citrate synthase, and we end up with the citrate molecule. And then in reaction 2, we have the isomerization of citrate to isocitrate. So again, same anatomical components, but just in a different configuration. And that's catalyzed by actinase, which have an iron sulfur center. So that. Okay. Next we have reaction 3, in which isocitrate is decarboxylated to alpha ketoglutarate. So remember, a carboxyl group is a COOH. So we're losing a COOH here in order to produce alpha ketoglutarate. The catalyzing enzyme is isocitrate dehydrogenase, which again makes sense because not only are we losing a carboxyl, also we have hydrogen from the isocitrate being transferred onto the NAD so that the NAD is reduced and isocitrate is oxidized. And this is a rate limiting step and the first NADH yielding reaction of the TCA cycle. So this is a dehydrogenase reaction that produces NADH that will go on to the electron transport chain. In reaction 4, the alpha ketoglutarate from the previous step is oxidatively decarboxylated to succinyl-CoA. by the enzyme alpha ketoglutarate dehydrogenase complex. And again, we have the production of NADH in addition to production of CO2. In reaction 5, we have the conversion of succinyl-CoA from the previous step to succinate, catalyzed by succinyl-CoA synthetase. And this involves the use of the high-energy thioester from succinyl-CoA to drive the synthesis of a high-energy nucleotide phosphate, GTP, through substrate-level phosphorylation. Okay, so it's able to add the phosphate onto GTP through substrate-level phosphorylation. And then GTP then undergoes conversion to I see. It undergoes a reaction with the enzyme nucleoside diphosphokinase in order to transfer the phosphate from GTP onto an ADP for ATP production. And then GDP is then resynthesized for continued availability of this step of succinyl-CoA to succinate. So we've talked about before, sometimes in some figures they just show ATP, but it's GTP which is then converted to ATP. Okay, next we have reaction 6, in which succinate is then oxidized to fumarate through the enzyme succinate dehydrogenase. So again, we're taking some of the hydrogens from succinate, thereby oxidizing it and putting the hydrogens onto the FAD to be then go on to the electron transport chain for energy production. Then we have in reaction 7, there's no figure here, but we have the hydration of fumarate to L-malate through the enzyme fumarase. In reaction 9, we have oxidation of malate to oxaloacetate, which again is an energy-producing reaction via this dehydrogenase, malate dehydrogenase enzyme. And then here I just have kind of the overall reactions of this of the TCA cycle. So we start with an acetyl-CoA, NAD, FAD, GDP, inorganic phosphate, and water. And then that is ultimately converted to carbon dioxide, NADH, FADH2, GTP, hydrogens, and CoAsh. And CoAsh is just the reduced radical of acetyl-CoA. Okay, so here we have an account of all of the energy produced. So I find this very helpful to say, like, okay, what are we doing? So in glycolysis, we have, we use 2 ATP, but we produce, let's see, we produce, sorry, I got confused for a second. Okay, so we, sorry, I got a little bit confused there for a second. So we are producing in glycolysis 10 ATP, total 10, but we use 2 ATP in the first stages of glycolysis, and therefore we have a net of 8 ATPs produced in glycolysis. And then we have the TCA cycle, which produces, which remember this, after we get to a certain stage in glycolysis, all of our reactions are occurring twice, okay? So this is showing that we'll produce 24 ATP per cycle of the TCA cycle, but then remember we're doubling it because for each mole of glucose, it breaks up into those 3-carbon subunits in glycolysis, and then everything is happening twice. So we double everything. So ultimately, in the end, we're going to produce for every mole of glucose, we're going to produce 38 moles of ATP, and then we're going to use 2 moles of ATP in this initial stage of glycolysis. So you can see that this investment is well worth it because we're only investing 2, but then we're ultimately producing 38 moles of ATP. So remember that when you're looking at this, I'm sure you do remember, but remember that the NADHs are equivalent to about 3 ATP, and the FADHs are equivalent to about 2 ATP. So in different sources, you may see the net amount of ATP produced per mole of glucose might be a little bit different. And the reason that is because different authors may use different amounts of, or I should say different equivalents of ATP for NADH or FADH2. So some might use 2.5 or 2.5 ATPs per NADH, or if we get really in detail, different researchers have found that, at different stages, they actually are producing, slightly different amounts of ATP. So for the purposes of this class, you just need to remember it produces 38 and uses 2, but you might see it different in different sources. Okay, so next we're going to talk about the hexomonophosphate shunt and pentose phosphate pathway. And these are a secondary pathway of glucose oxidation. Now, generally speaking, we have different types of shunts in our bodies, and the purpose of these shunts is to generate intermediates that we need for production of certain important molecules that are not produced in other pathways. So our body has utilized the hexomonophosphate shunt and pentose phosphate pathway to produce intermediates that we need for synthesis of certain molecules that are not produced in other pathways. So that's shunts in general, but the hexomonophosphate shunt and pentose phosphate pathway have a few very specific functions. So the first function is to generate reducing equivalents, NADPH, for fatty acid and steroid biosynthesis. This molecule also contributes to the maintenance of reducing substrates in red blood cells to ensure functional integrity, and the NADPH is also utilized for drug metabolism. In addition to producing NADPH via these pathways, we also get the production of 5-carbon sugars, the ribose 5-phosphate, that's needed for the synthesis of nucleotides and nucleic acids that are absolutely essential for DNA and RNA synthesis. And then also this pathway allows carbon dioxide to be released outside of the mitochondria. Okay, so this pathway, the HMPS-PPP pathway, occurs in the cells of the liver, adipose tissue, adrenal cortex, testes, lactating mammary glands. Okay, so this really makes sense when we think about what this pathway is doing. So fatty acid, it's producing NADPH that is ultimately going to lead to fatty acid and steroid synthesis, okay? So all of these tissues, that's really important for. Testes are important for steroid synthesis, lactating mammary gland and adipose tissue are important for fatty acid synthesis, and so on. And this pathway operates exclusively in the cytoplasm, so fatty acid synthesis also takes place in the cytoplasm. But we have a low activity of this pathway in skeletal muscles because they have a limited demand for NADPH for fatty acid synthesis, and also because they just have such high needs for energy and rely on both glucose and fatty acids for energy rather than production of these intermediate substances, the NADPH and the ribose 5-phosphate. So actually 30% of our glucose oxidation in the liver occurs via the pentose phosphate pathway. And finally, the conversion of ribonucleotides to deoxyribonucleotides, so the nucleotides from RNA to DNA, also requires this NADPH. Okay. So we have two stages of the hexomonophosphate shunt and pentose phosphate pathway, but they're coupled. There's an oxidative arm and a non-oxidative arm. So both these figures have the whole pathway, but are emphasizing the different arms of the pathway. Okay, so first we have the oxidative stage, where our initial substrate is G6P, and it occurs at the beginning of the pathway. And it generates 1 mole of NADPH for every mole of G6P that enters the pentose phosphate pathway. And this is catalyzed by G6P dehydrogenase. So again, this makes sense. We have the NADP. It's reduced by taking the hydrogens from the G6P to produce NADPH. So this is G6P dehydrogenase. And then we have the second step, which is the 6-phosphoglutamate dehydrogenase, again producing NADPH by oxidizing the 6-phosphoglutamate to ribose 5-phosphate. Okay. And then we ultimately have the production of the ribose 5-phosphate. So you can see in this oxidative phase, we produce both the NADPH that we need for the functions that we talked about earlier, including fatty acid and steroid biosynthesis. And we also have production of this ribose 5-phosphate for DNA and RNA. Okay, we also have the non-oxidative arm. So here they're showing the oxidative pathway just with this kind of dotted line. So this dotted line is demonstrating everything we see here in this oxidative pathway. But in this pathway, I won't go into a whole lot of detail, but in the non-oxidative stage, we're converting the 5-carbon sugars into F6P, okay, and into glyceraldehyde 3P. So we're converting it into 6-carbon and 3-carbon sugars that can then enter the glycolytic pathway, okay? So here we have production of R5P, and then in the non-oxidative form, we're regenerating these intermediates, F6P and G3P, that can then go into the glycolytic pathway to be further oxidized for energy. And the primary enzymes of this non-oxidative stage are transaldolase here and transketolase here. And these enzymes require thiamine pyrophosphate as a cofactor. So you can just, the only thing you need to remember of that is that thiamine is a cofactor in this pathway. Okay, so we talked about glycolysis. Now we're going to switch gears and talk about gluconeogenesis. So this is the biosynthesis of new glucose from other substrates. And gluconeogenesis is necessary for tissues that rely heavily on glucose and can't utilize other substrates for energy, or have very limited ability to do so, such as the brain. So these tissues include the brain, testes, erythrocytes, and kidney medulla. So these tissues need the actual glucose in order to, as a fuel source in order to function. So if they don't have the actual glucose, then we need to produce new glucose so that they continue to have a fuel source. And as I said here, the brain can derive energy from ketone bodies, but it's not desirable because unless you have a very specific condition, the ketone bodies are going to lead to ketoacidosis, which is very harmful. Now, gluconeogenesis, we should be able to go through this pathway pretty quick because it's essentially the reverse of glycolysis. Oh, I just noticed that you guys aren't. Sorry, I didn't notice that you had my whole screen on there. Okay, so this should be better. Okay, so here, this is a very busy slide, but it's showing something that you should be able to grasp pretty easily after our last conversation. So here in this figure, I like it because it's showing on the left here all of our steps in glycolysis, okay? So the conversion of glucose to pyruvate. And on the right here, it's showing gluconeogenesis. So what this figure is demonstrating is that the gluconeogenesis has all the same steps, but we're just doing them in reverse. So we're synthesizing glucose from pyruvate, 1 glucose from 2 pyruvate. Now, the interesting part of this, so here we kind of show it upside down. So this is just the gluconeogenesis moving from pyruvate to glucose. But gluconeogenesis from 2 pyruvates up until this stage here of the 1,3-biphosphoglycerate actually consumes 6 ATP, okay? So that's important to remember. When we're trying to produce glucose via gluconeogenesis, we actually have to use 6 ATP. So this makes it really costly energetically, considering that glycolysis of 2 moles of pyruvate only really yields 2 ATP, okay? So this is very costly energetically, and we really only do it because we need to supply tissues that depend only on glucose with glucose. Okay, so we already talked about that. Okay, in the liver and in the kidney cortex, G6P that is produced from gluconeogenesis can be incorporated into glycogen, or the glucose can be released in the blood, okay? So we talked about in the liver as compared to the skeletal muscle, which does not have G6P phosphatase, the liver and kidney are able to convert G6P to glucose to be released in the blood, or they can use it to incorporate into glycogen. So in the muscles, we can't really release the glucose into the blood, but it can be G6P can be utilized to build glycogen. Okay, so the whole cycle that we just went over. Oh wait. Okay, did I skip it? Okay, I skipped it. Or I skipped a slide. Okay, so now I want to talk about the substrates that we can use for gluconeogenesis, okay? So lactate, which remember where we get lactate? We get lactate when we anaerobically convert, when we have anaerobic conditions, and then we convert pyruvate to lactate, okay? And lactate is the predominant source of carbon atoms in gluconeogenesis, okay? So during anaerobic glycolysis, the skeletal muscles and erythrocytes primarily reduce pyruvate to lactate. That they remember erythrocytes don't have the ability to for oxidative phosphorylation, and skeletal muscles do not have the ability for oxidative phosphorylation if we just don't have enough oxygen, such as in anaerobic conditions. like very heavy exercise. Then, once lactate is produced, it's released into the bloodstream and transported to the liver for gluconeogenesis. The glucose produced from the gluconeogenesis in the liver is then returned to the blood for use in the muscle as either an energy source or to replete glycogen stores. So in this way, the liver can convert an anaerobic byproduct of glycolysis, lactate, back into glucose to be reused for non-hepatic tissues. And this cycle is called the Cori cycle. I have a graphic of that right here. So this one is showing the muscle versus an RBC. But here we have glucose is converted to pyruvate via glycolysis, but then in anaerobic conditions, the pyruvate cannot go through the TCA cycle and electron transport chain. So instead, it is converted to lactate by lactate dehydrogenase. The lactate then is transported out of the cell into the blood and goes to the liver, where the liver is then able to use the lactate as a substrate for gluconeogenesis, and then the glucose can go back to the muscle to be reused, either to go back to lactate if there's still anaerobic conditions, or it can be oxidized for energy, or used to synthesize glycogen. Okay, so our next substrate for gluconeogenesis is pyruvate. So pyruvate is generated in the muscle and other peripheral tissues. It can be transaminated to alanine and returned to the liver for gluconeogenesis. So remember, transamination means that an amino group is transferred between two molecules, okay? And then, so this transamination requires an amino acid as a donor of the amino group and creates an alpha keto acid. And this pathway is called the glucose alanine cycle, okay? So when alanine is produced via transamination, it goes back to the liver, and then it can be converted, it can be deaminated to be converted back to pyruvate, which is then used for gluconeogenesis or oxidation via the TCA cycle. Then the amino nitrogen is then converted to urea and excreted by the kidneys. Okay, so the major function of the glucose alanine cycle that utilizes pyruvate as a substrate for gluconeogenesis is that it allows non-hepatic tissues to deliver the amino portion of catabolized amino acids to the liver to be excreted as urea. So let's look at this graphic here, okay? So we have the muscle, we have our glucose in the muscle, okay? It undergoes glycolysis to form pyruvate. We have transamination here, okay, to produce alanine. So we can see the input of a nitrogenous compound here, and we have production of alanine. Alanine then goes into the blood and back into the liver, where we have deamination. So here we combined the amino group with the pyruvate to produce alanine, but then once we get to the liver, we're going to have deamination, where this amino group is taken off. The amino group then undergoes conversion to urea to be excreted by the kidneys, okay? So it's taking kind of this really almost this waste product and making and allowing us to excrete it via the kidneys. And then we have the pyruvate left here, which can then undergo gluconeogenesis to glucose, and then the glucose can go back to the muscle cell to be incorporated into glycogen or to be oxidized for energy. Okay, and this figure is showing a combination of the Cori cycle and the glucose alanine cycle and just kind of putting it all together. So let's kind of just start here with the muscle where we have glycolysis of glucose into pyruvate. Okay, let's start with the Cori cycle. Then we have conversion to lactate in anaerobic conditions. The lactate goes through the blood back to the liver, and then we have reconversion of pyruvate and ultimately glucose that can be transported out to other tissues. We also could have the pyruvate in the muscle be converted to alanine through transamination using the products of protein degradation. The alanine then goes back to the liver to reproduce pyruvate, and again, then we can undergo gluconeogenesis, and the glucose can be utilized by either the liver itself or peripheral tissues. Okay, so then we have our next substrate that we'll talk about for gluconeogenesis is amino acids, and nearly every amino acid can be degraded to an intermediate of the TCA cycle, except for leucine and lysine. So when we talk about which macronutrients these amino acids can be converted into, we use the terms glucogenic, so they can be converted into intermediates of the TCA cycle, or we use the term ketogenic, which means that they can be utilized for fatty acid synthesis. And just the leucine and the lysine are strictly ketogenic, and all of the rest of the amino acids are at least somewhat glucogenic, though some of them can be both ketogenic and glucogenic. So when the amino acids are degraded, we can use the carbon skeletons of the amino acids to be converted to oxaloacetate, which can then be converted to pyruvate, which we can use for gluconeogenesis. So in the last slide, we talked about the amino group of amino acids being used for the glucose alanine cycle, but in this slide, we're talking about the carbon skeletons of the amino acids that are then converted to oxaloacetate for ultimate fate of gluconeogenesis. Now, when glycogen stores are depleted in the muscle, we have catabolism of muscle protein, and this is where we get the amino acids that contribute to gluconeogenesis that we use as a major source for blood glucose maintenance. So it's a little bit tricky because we say that muscle, in the skeletal muscle, we can't have actual production of glucose itself, only G6P, because we're lacking G6 phosphatase, but degradation of the amino acids in the protein yields these carbon skeletons that we actually can ultimately use for gluconeogenesis in the liver in order to maintain our blood glucose homeostasis. We can also utilize glycerol for gluconeogenesis. So the 2-carbon unit of acetyl-CoA can be derived from beta oxidation of fatty acids, and that can be incorporated into the TCA cycle. However, in the TCA cycle, we have that 2 carbons are lost as CO2. So really, when fatty acids are degraded as an intermediate to the TCA cycle, we don't really have any net conversion to carbohydrate. But the glycerol backbone of lipids can be used for gluconeogenesis, and this requires phosphorylation of G3P by glycerol kinase and dehydrogenation by G3P dehydrogenase. Okay, and then our last substrate that we can utilize for gluconeogenesis is propionate. So propionyl-CoA is a terminal generation product when we oxidize either fatty acids that have an odd number of carbon atoms or when we oxidize certain amino acids. And the propionyl-CoA can be converted to succinyl-CoA by an ATP requiring enzyme, propionyl-CoA carboxylase. But there's actually a number of steps in this process where we convert propionyl-CoA to succinyl-CoA. One of these enzymes uses vitamin B12 as a cofactor. And then, of course, once propionyl-CoA is converted to succinyl-CoA, then it can enter the TCA cycle at that step. And here is our last slide, which are the practice questions that you can use to test your knowledge of this. And this is the only topic that we're going to discuss today. It is kind of a big, dense one. So if you have any questions, of course, you can always contact me, and have a great week.